Yes, perfect. Thank you so much, everybody, for joining us. Thank you, Lee. And welcome. So we're very happy to have you all here today for this webinar hosted by the British Occupational Hygiene Society. If you're joining us as someone maybe a little less familiar with the BOHS, we are the Chartered Society for Worker Health Protection, and we're the proud home for occupational hygienists and other associated professionals. And we often hold webinars and meetings on topics of interest to occupational hygienists, though today we're very fortunate because we have a very embracing subject, as the Exposome is a concept that is important to all professionals who are engaged in occupational health. So we have three wonderful speakers today. First up will be our BOHS President-elect, Param Gahir. As President-elect, Parm will be touring the UK regions to introduce himself and his upcoming presidential theme at these BOHS meetings. So today gives Parm a nice big tick for his tour, although today I suppose it's more of a virtual tick because it's a webinar. And so Parm will present for around 15 minutes or so, and then after that we'll go straight into the Exposome content, which is led by Miranda Lowe and Manosa Jagash, who work together on the E4 project. So yeah, as we go, please do put your questions, comments in the chat box. I'll be keeping track of it as the webinar goes on, and we'll have that Q&A at the end. So over to you.
Thank you very much, Mary. Let me get 2 seconds and get my presentation up on the screen. Here we go. Can we all see that? Mary,
yep,
can you
see that? Yep, we're good for that. If you just full screen
perfect.
that, and we're good to go.
Okay, let me just get back onto that again. I've messed it up. Sorry. I pressed the wrong button. Apologies, folks. Here we go. Can you all see that now?
Oh, wrong one.
Can you see that, Mary?
We're seeing your Teams invite, but I think you might have shared the wrong screen.
Let me go again. Apologies, folks. I did have it right. Let me start again. Third time lucky. Stop sharing. Start this one. Go here. President-elect. Can we see that now?
Good. Yep.
Excellent. Right. Apologies for my poor IT management there, folks, this morning, and many thanks to Mary and Lee for the introduction this morning. This presentation is something that I put together as President-elect for the BOHS. We're encouraged to come up with a theme that we then drive and promote through the presidential year. And I personally struggled to find something that I could talk about that stands out, that's a little bit different, hasn't been done before, and provides something for you guys to think about, as well as for me to think about. And what I'm going to do is I'm going to spend 15 minutes sitting on the fence this afternoon and casting my mind back to how I started as an occupational hygienist and where we currently are and where we might be in the future. So the title of my presentation this morning, or this afternoon even, I'm making the same mistake, is Back to Basics, Embracing the Future. So a little bit about me for those that don't know me, and I'll flick through these quite quickly. Currently, I'm fortunate enough to be the head of occupational hygiene for a consultancy business in the private sector. Previous to that, I've been a HSE specialist inspector of occupational hygiene for nearly 7 years. And overall, I've got 35 years experience as being an occupational hygienist. I'm a bit unique in that I didn't fall into this. I've got a degree in occupational hygiene far too many years ago, 35 years ago. Crikey, I'm old. So I deliver this talk for real, and I appreciate it's going to be a little bit different to do this online. But my first question is, what do occupational hygienists do? Now, with the limitations of working online, I'm going to ask the question and then answer it myself as well. So if I put this slide up here, what do we do as occupational hygienists? We're traveling in a time machine, looking at things that have happened in the past or currently happening, but we still need to remain in the time machine and then tie that information up with what could happen in the future. So what is the what could that we're worried about? We're worried about the health impact on people that are employed and work in factories and other environments where they're potentially exposed. Okay, so what's a time machine got to do with occupational hygiene, you might ask? So I put together a couple more slides here, okay, and let's look at this back as far as time will allow us, and my reading and research allows us to understand. So this chap here, Agricola, okay, a Greek philosopher, because they weren't really called scientists at the time, he discovered lead poisoning in miners. So just hold that thought there for a moment. Then we get to the Romans, okay, cinnabar crushing. They used that as a dye. Mercury sulfide and vermilion, they were all used to create the vivid colours in some of the clothing that they wore. They only had a couple of different colours that they could dye their clothes in, but people, as far as the 1st century, were noting that things go wrong. If you do a job and you inhale or are exposed to something, it will have an impact on the human body, and that impact was beginning to be noted. That resulted in the 1st century the good old goat coming into play. And again, I would ask the audience, what's the relevance of the goat here? So again, I lose the impact a little bit when presenting online like this, but the purpose of the goat was it was the first bit of PPE that was ever developed, okay? The bladder from the goat was taken and it was used as the earliest recorded form of respiratory protective equipment that we can find in any documented writings or recordings. Okay, so still think about my first slide, the time machine, and think about what I'm presenting here. We've got problems through time that people are seeing. Okay, we then get to the 16th century. Okay, and we get Ramazzini, who's now actually thinking about how to control the exposure that's occurring, to prevent, manage and limit the ill health and harm that's occurring. He documented that a wet cloth or some material soaked in water around the face will provide some degree of protection. I would say in the 16th century, that was groundbreaking and novel they'd come up with. It's the first time that we've got documented writing anywhere that somebody is not only thinking about hazards and how they manifest themselves, but we're now beginning to think about control. So what did Ramazzini put together? Okay, he put together this book, De Morbis Artificum, and this is his documented writings of some of the diseases that he saw while he was conducting his work with groups of people. Now, this is the list from the 16th century taken from his book. Now, look at that in modern speak and how we work and what our understanding of occupational hygiene is. Musculoskeletal disorders, we're all familiar with those. Stress-related health conditions, perhaps less so as occupational hygienists. My training certainly doesn't extend me to understand those with any degree of competence, but certainly I'm beginning to appreciate that that's something that we need to think about. What else has he seen in the 16th century? An increase of breast cancer in nuns. No explanation as to why, but he's seeing, okay? He's talking about sensory systems diseases, okay? These are people making hats exposed to mercury vapor while they're pressing animal pelts, okay? Familiar. Impact from environmental pollution, people living near dirty workplaces. Leather tanning factories was something that he documented, and the impact it has on the locale and the environment around it. Okay, and he's also then talked about inequality and vulnerability, and here he's looking at young people. It was quite common for children to work in factories and women to work in factories who often weren't looked after in the same way as the men were. So can we begin to see a little bit of familiarity of what happened in the 16th century to where we currently are now with the work that we do? So, this has been around for a very, very, very long time. So what is the point of me bringing this up? What's the point of this history lesson here? Okay, how was any of this possible? Did any of these Greek scholars and scientists and physicians and other clever people, did they have any of what we're used to in the 21st century? Okay, did they have any guidance notes? Did they have any legislation? Did they have the British Occupational Hygiene Society to call on? Did they have the regulator to ask questions? Did they have any technical or scientific equipment? And the simple answer is no. Okay, they didn't have any of this equipment. Okay, so the question begs, what did they do to arrive at these, what I would say, well-documented and now well-respected positions about workplace or environmental hazards and the impact that it has on worker health protection? So how have these guys got here? And there is a question here that I'm going to answer in a little while. Okay, so what are we doing now? Okay, evolution. Let's look at what we do now. When I first started my career as an occupational hygienist, we used a B&K sound meter like the one on the left. It took 8 D-cell batteries, and you needed to have 3 shredded wheat twice a day to be able to pick that up and walk around the factory with it. But look at it now, the instrument on the right hand side, it's another B&K instrument, the 2250, and that meter has probably got more computing power than my first ever laptop computer that I bought when I was at university. But is that really the thing that's solving our problems? The Greek philosophers and the Roman scientists and all those clever people 3, 4, 5, 6, 700 years ago, did they have any of this technology? Did they have any technology at all to allow them to do what they were doing? This was the question that was going around in my mind. Okay, what was it that they've done that's helped them arrive at a position which we now, hundreds of years later on, respect for being reasonably accurate within the limitations of what they knew at the time? So the next slide, okay, are we making any progress in this? Okay, and again here, this is designed to be an interactive slide where I would ask the audience, and again, it's a tad limiting with a virtual setup like this, but I will answer my own questions here. Okay, are we making any progress? Okay, I would say we are, but there's limitations in that. Okay, so casting my mind back to being a regulator, where I spent a lot of time looking at the work produced by other people, some of the common themes that we were seeing, that there was no explanation. Now, what do I mean by that? You'd pick up a report and you don't know if you're in a brickwork, in a food factory, in a flour mill, or anywhere else. You've just got numbers and data. So without that contextual explanation, it's very difficult to put yourself into the mindset of that piece of work that you're looking at. Okay, often there's no justification or reasoned consideration. Okay, so what do I mean by that? What have you done and why? Okay, what were your answers, and how have you put that in the pot, stirred it around, and got something useful at the end of it? Think about making a pot of soup or a dish or something like that. You've got to have steps in the process to introduce the various different elements in the right way for the correct reasons to get the desired end outcome. We don't see that. Okay, we also see sometimes a lack of understanding of the whole thing that we're looking at. Okay, but again, you might be thinking, why am I taking you here? Why is my talk themed back to basics, embracing the future? And I've talked about some scientists from several hundred years ago. We've introduced some of the problems that we're seeing now, and I've shown you two random pictures of a very old and a very new bit of noise equipment. There is method in my madness here, okay? So why is my next question? And again, I lose the impact of the interactiveness when working online like this. But let me take you back to my beginning, okay? That was me on the left-hand side, had a bit more hair then, and looks like my trouser size was a couple of sizes a bit smaller. And to the right is Mary should be able to recognise who he is, and Lee and perhaps a few others, but that's my good friend and ex-colleague Duncan Smith. He and I were at university together, and he now currently heads up the health unit at the HSE, and I had the privilege of calling him my boss as well as my good friend for 6.5 years. But you're still wondering why. What is the relevance of all of this? Back to basics, embrace the future. Well, it was while I was at university that I had the most important lesson in my life as an occupational hygienist, and it's something that I work with and cherish to this day. And again, I would ask the audience, what do you think that lesson is? We're unable to do that, so I'm just going to give you the answer here, okay? And it's a chap called Robin Bryce. Robin Bryce was my coach and mentor, and he was the chap that ran the occupational hygiene department for Boots UK at their manufacturing site in Nottingham. I spent 18 months in a placement year from university learning how to be an occupational hygienist in the real world, in a real factory where they had real problems of many, many different sorts. So again, embrace the future, back to basics, embrace the future. What did Robin teach me? Okay, he taught me two things. And the first one that he taught me was to look, to see, and to ask questions. Okay, he said the best occupational hygienist is the one that looks at a situation, stops, thinks, evaluates, and asks. Okay, you don't need any technical equipment, you don't need anything scientific, you don't need any fancy gadgets and gizmos, you don't need the latest Bluetooth, all singing, Wi-Fi enabled, whatever. You just need to turn up and to look and to ask questions. Understand what's going on. Okay. The second thing that he said that you need is a notebook, because it doesn't matter how good you are, you're never going to be able to remember everything. And he taught me that once you've looked, you've seen, you've asked, you've listened, write it all down. 35 years later, I still work in the same fashion, where everything starts with me looking, seeing, and asking questions, and everything concludes by writing things down. Okay, I've still got every one of my notebooks from the last 35 years that I've ever made any occupational hygiene notes in. So again, I throw it out there, back to basics, embrace the future. And all I've done is introduce some random Greek scientists. I've told you some problems that we're experiencing, and I've highlighted a little bit about me going to university and some random chap called Robin Bryce that you've never heard of. So let's start joining some of this together, okay? How does this work? So what does the future hold? Now, let's start right at the very beginning. What did these Greek scientists do? Okay, they didn't have any technical equipment or instruments. They didn't have any guidance documents. They didn't have any legislation. They didn't have any phone a friend options. They didn't have the BOHS or any other society that promotes and endorses occupational hygiene. They didn't have the regulator to call on. What did they do? They went in, they looked, they listened, they saw, and they wrote things down. What did Robin Bryce ask me to do? He told me to slow down, stop, think, look, listen, and record. So what impact does this have on our future? Okay, we've got artificial intelligence coming in now. Okay, there's instruments that can think for themselves. There's sampling pumps that can turn themselves on and off whenever you want them. They can tell you how accurately they've made flow and calibration. Okay, there's instruments that can talk to other instruments that can tie in. Okay, we've got stuff that's moved from analog technology with dials and needles that move to digital readouts. Is one more accurate than the other? Not necessarily. Okay, we've got robotics coming in, we've got automation. There's tasks that people can easily be replaced with. Okay, and then we've got the last thing, Chat GPT. We've got some clever software that can probably, not tried it myself, but probably almost put together a passable occupational hygiene report. Okay, there have been legal cases where I'm aware of one solicitor or barrister that's used Chat GPT to put together his legal case that got found out. So we're at that boundary, we're at that sort of cutting edge junction in what we've done previously to what we need to do moving forwards. Now, what is my conclusion with this? Okay, back to basics and embracing the future. And this is one of my most favorite slides. Okay. And again, I would ask the audience to interact. Mary, this is my last slide now. Occupational, I challenge you to think about what we do totally different. Okay, what I say and how I think is occupational hygienists are a type of doctor. You can go to your medical GP and present with a rash on your skin or your arm. The GP can look at that and say, yep, it's this. Or the GP says, no, not quite sure what it is. He'll phone a friend, look in a book, consult Dr. Google and arrive at a diagnosis. But we're different. We are looking at a range of circumstances and situations. They may or may not do something. There's several factors that we apply into the evaluation, and then we're predicting what might happen in the future, and we're making that prediction of it's likely to be good because you're controlling the risk well, or it's likely to have a detrimental outcome because of an X, Y, Z, a number of reasons. So how does that tie into back to basics and embracing the future? For me personally, sitting on the fence, we need to have one foot firmly in the past, where we mustn't forget our ability to talk, listen, see, understand, engage, good old-fashioned pen, paper, torch, glasses, eyes, look and see. Then we need to write things down to make sure that we remember and document things correctly. But equally, we mustn't be afraid of the future and embrace the modern technology that's coming in that allows us not to make the decision, but allows us to make refined decisions in the work that we do as occupational hygienists. So folks, a little bit rushed from me this morning. I would take much longer and I would engage with the audience a bit more. But for me, in my presidential year, I encourage you all to go back to grassroots, think, listen, act, do, ask, see, write things down, but also be forward enough to see how the modern technology that's coming into the world of what we do, how is that not going to replace what we do, but how's that going to enhance what we do? Thank you, Mary.
Thank you. Perfect. Thank you so much, Parm. Yeah, very, very interesting theme for your presidential year. And I find it's going to tie in really well to this webinar. We're embracing the future here. What does Exposome have to hold for us in the future, right? So we're going to hand over next to Miranda. Miranda is the Director of Scientific Growth, Engagement, and Innovation at IOM. So she'll be presenting first on methods for characterizing the external exposome. And then she's going to hand over to Manosic, who is the senior postdoctoral researcher at the Center for Environment and Health at KU Leuven in Belgium, and he'll be presenting on methods for characterizing the internal exposome. So I'm going to hand right over to Miranda. You take it from here. Oh, where'd you go?
I see Miranda is frozen, the screen at least.
Oh no! Oh, what happened? Uh-oh.
I think I'm back.
Oh,
yeah, there you are.
Am I back?
Yeah,
we can hear you.
Yeah.
Okay, the minute I think you started speaking, Mary, it just kind of shut down. So hopefully it won't happen again.
Oh no. Can you share your slides okay?
Yeah, it should be okay. Let me try. Hang on a second, okay? I think perhaps my computer was so overwhelmed with what Hamzat had to say.
It's just too much.
Couldn't take it. Hang on a second. Let me just get my slides ready. Oops. Right, I'm going to share my screen. If you bear with me a moment. Can you see my slide?
Yeah, looking good.
All right, I'm going to try the presentation mode.
Perfect.
Can you see that?
Yeah. It's not gone big yet.
Okay, hang on. Let me see if I can change what I'm sharing.
Okay, that's perfect. Yeah,
yep.
It's great.
Is that right? Okay,
off
good.
You go. Yeah, thank you.
Okay, so thanks very much for inviting myself and my colleague Manosic here to discuss a project that we're both working on. It's called the Exposome Project for Health and Research, or E4. And I'm going to first start with a little bit of information about what the exposome is. So you may or may not have heard of this term before, but you probably have heard of the genome. So the genome is essentially all of your genes, and we have the technology now to be able to actually map out the genome and look at where specific genes are, and this has really helped a lot in terms of our ability to research disease and causes of disease. But we also know that genes only explain part of how disease comes about, and we're talking mostly about chronic disease here. So there is this concept called the exposome. So we know that the environment interacts with our genes to actually then result in a manifestation of disease. And we know that in studies where identical twins who have exactly the same genome are raised or live in different environments, they may have different disease outcomes. So this concept then is, if we can map the human genome, can we then do a similar mapping exercise for the environment that we live in? And the environment can be very broad. So it may be things like occupational hazards, smoking, our diet, air pollution, but it can also include the types of environments that we live in, whether it's urban environments, rural environment, climate has an impact, and also our social environments have an impact on our health. And so we take this idea of environment quite broadly as the external environment, but then the exposome also looks at how that external environment interacts with our internal physiological environment to then result in some kind of health impact. So that's a very big concept, and there's been a lot of research in the last couple decades looking at this concept, and it probably will not be something very uniform like the genome. But we're looking, and I'll talk a little bit about the approach we're taking in this project, which is similar to approaches taken in other exposome projects. So E4 looks at the working life exposome. The concept of the exposome has primarily been applied in environmental health. It is now also being applied in occupational health. But we chose to call, rather than occupational exposome, we actually are looking at the working life exposome, because we know that work and health work and life are a continuum. So your work will impact your life in general, as well as the other way around. So we want to look at the working life and health relationships. We want to understand a bit more about what are vulnerable life stages and populations. And what essentially this project is hoping to lay the groundwork for is better prevention that's evidence-based and cost-effective. So this is a Horizon 2020 project. So it's funded through the European Commission. And it's a very large project, including 19 partners from 12 countries. So there's myself from IOM here in the UK, and Lenosage is from the Catholic University of Leuven in Belgium. And we have many other partners around Europe. So why, again, do we want to have a project such as E4, looking at the working life exposome? So we know that occupational disease in European countries account for about 5 to 7% of mortality, so deaths, and this can cost 2 to 6% of GDP. And potentially the burden could be greater depending on the methods that you use. And we know that again, as I mentioned, work and life are not so indistinguishable. And so we want to better understand that relationship. And to do this, we need to move from the traditional occupational epidemiology or occupational health research approach, which looks at usually a single occupational exposure, linking it to a single health outcome or disease outcome, to really looking more holistically at exposures and disease. We also want to be able to better understand vulnerability. So what are the different impacts on different groups, as well as we want a better understanding of the biological mechanisms of disease. And this is very timely because we are facing a lot of challenges coming up, such as demographic challenges. So we are all working longer. Retirement age seems to be increasing. And also there's kind of more female participation and generally kind of a more of a gender awareness within the workplace. And also the nature of work has changed. So we're looking at different contracts of work. People don't necessarily work in the same industry all their lives anymore. So the types of cohorts we used to have, which are kind of industry-based cohorts, they may need to change. There is a lot of automation in work now. Prompted mentioned things like AI and robotics. So how are we going to look at this ever-changing nature of work? And so again, I mentioned before, the exposome is something that had been primarily applied in environmental health projects. And what hasn't been looked at so specifically is the working life or occupational side of things, which is what we're doing. So one of the outcomes we hope to have in this project is to have a toolbox that's online, which includes the various methodologies that we're developing within the project, some of which we'll be describing today, as well as results of the research. We are aiming to provide tools that can be used by other scientists, by policymakers, as well as occupational health practitioners such as yourselves at BOHS. And some of you may have also participated in other initiatives we've done on this project to help engage practitioners in our work. This is a little bit about what happens in the project at a very high level. And it's quite comprehensive. So we are doing a mega cohort, which is an epidemiological study where we pool together many cohorts across the EU. And this increases our power, and basically power means the number of people in our studies, so we can better look at the impact of working life risk factors and non-communicable diseases, as well as I mentioned before, the vulnerable life periods or subgroups of the population. It helps to have very large numbers to do this. We will also be looking at a couple of case studies which focus on respiratory disease and shift work. And this is relevant for the work that Nosage and I are doing in terms of applying some of the methodologies we describe in shorter term studies. So there is both an external exposure part of this as well as an internal exposure looking at biomarkers and biological pathways. So all of this are different parts of the project. And we're also looking to develop tools on how we can translate this into health and economic impact assessment. So it's very comprehensive. As I mentioned, again, all of these will be, the outcomes of this project will be placed into the toolbox. And in the interest of this, I'll just move on now to the external exposome. Please do, if you have any questions about the project as a whole, we're happy to answer them later on. So we decided to focus this webinar on two aspects of the E4 project. So I mentioned very many. We have the cohort studies, we have health impact assessment studies, but what we thought we would focus on in this case are the measurement methods that we're using to quantify the external exposome. And so the external exposome is traditionally what a lot of us think about as exposure. So how we come into contact with inhalation contaminants, dermal, etc. And then Nosage will talk about our approach to the internal exposome. So the external exposome is it's a work package that that takes place in E4. It's led by myself at IOM, and we work very closely with our partners at TNO, and you can see the main members of the team on this slide. So a brief introduction. Traditional exposure measurement, so that's what a lot of you will do, as well as myself in the past. So how convenient is it? We often have multiple samplers and pumps and different devices, and this is just an example. It's not an occupational study, but you can see that if we wanted to measure multiple exposures, which is one of the things we want to do in this study, you have to have quite many different samplers that you would hang on a person. So we kind of want to move away from this Christmas tree approach as much as possible and consolidate that. And so we want to look at tools that will enable us to measure multiple exposures in a more efficient and effective way. The other aspect of the exposome is that it's looking at exposures across time. And the exposome is actually defined as all your exposures across your lifetime from conception till death, and which is a very large concept. So we're, in this case, capturing a bit of a snapshot in time, but we also need new technologies that can help us provide a more complete and integrated overview of an individual's exposure situation or exposure profile. And so again, as I mentioned, this particular package is based on, it's looking at developing methods for characterizing the external exposome. So these methods are going to be applied or are being applied right now in two different case studies. One of them is focused on looking at respiratory health and the other at shift work. This respiratory health study is based on some existing cohorts in Europe, and I will focus more on this part of the short-term aspect of the case study. So they will be applying, they will be applying the methods that we describe in a subset of the population over about a week's period of time. So the measurement methods will be deployed for about a week, and of interest, the health outcomes that we hope to be able to look at and link exposures to include lung function, respiratory symptoms, and affect biomarkers. And specifically, we're interested in looking among asthmatics. In the shift work project, we are interested in looking at short-term outcomes related to cardiovascular health as well as aging. Sorry, my slide's not advancing, I know. Okay, so what's our approach to exposure assessment? So we are, as I mentioned, we want to measure multiple exposures. So we've developed a number of methods that are fairly small for them, the participants to be able to carry, but we then have to integrate all these methods together. So we have an environmental sensor, which a participant can wear over the week-long period. We also have a novel passive sampler, which is geared at measuring chemical exposures. We have a questionnaire that participants can fill out on their phone, and this is using something called the ecological momentary assessment method, which is a method where we have set questions that can be pinged to a participant at different times of the day. And these include things, they're slightly different between the case studies, but for the respiratory health one, some of the questions are around symptoms. We also have questions about when do they go to work, how do they commute to work. The shift work one has questions about diet. So you see that it's kind of tailored to the questions that we're interested in, but it also helps us gather some contextual data about the participants' lives. We're also looking at having a heart rate monitor, for the shift work study, and a physical activity monitor for the respiratory study, as well as the heart rate monitor will also do physical activity. We also, in the homes of some of the participants, we will be doing passive area sampling for biological exposures. And in terms of the health outcome related data for the respiratory study, lung function is being taken. We'll also be taking blood and urine samples, as well as exhaled breath condensate in some of the participants. So lots of things to measure. I'll go through some of the main ones primarily for the external exposome. So the methods that we're using to measure the external exposome include the wearable sensor system I mentioned. And if you remember before, if you want to measure multiple exposures, you've got to have lots of different devices. Using some of these smaller sensor technologies, we're using a sensor box, which looks like the one you see here. It's quite small, and it measures UV exposure, light, dust or particles, noise, temperature, humidity, sleep. It doesn't measure sleep. Physical activity and heart rate, that's done through separate monitors. As I mentioned, we have the phone app, which is a questionnaire. And the passive sampling method for chemicals I mentioned is a wearable combined 10x and PDMS or silicon sampler. And you can see a picture of what it looks like there. It's a similar size as, for those of you familiar with charcoal tubes, it's like a size of a small pen that a person can clip on to their lapel or to their clothing. And for the biological samplers, we're using an electrostatic dust sheet, which you can see here on the bottom. And it's laid out for about a week-long period in a person's home to capture basically the dust, and then that's analyzed for microbial diversity. And currently, these methods are all being implemented in the 2 case studies I mentioned previously. So that's a lot of data. What are we going to do with it? So these technologies provide new information. So personalized, much of the data is personal. So it can give us an assessment of individuals' exposure. The sensor data is high resolution data. So this is time resolved. So it's minute by minute data, which could be potentially linked to acute health effects or short-term health effects. And it can also help us investigate the determinants of exposure. Because we're measuring multiple exposures, we can look at how they relate to each other. And what we hope to be able to do is then be able to give each person an exposome profile, and then to see what are the commonalities between these profiles. Of course, this brings challenges. So there's a lot of data, and therefore we need more elaborate data processing methods. And there are relatively few methods so far that have been developed for this type of data, but it's ongoing and being developed all the time. And of course, how do we prioritize between exposures? So what are we doing? There's a lot of data and there's a lot of QA, QC and data processing. And this is just a hint of the kind of things that we're doing. We have a lot of processing of the high resolution data. We have to make sure it's complete. There's enough data there. What are we going to do about missing data? And for those of you, of course, hygienists are very experienced in data measurement. There's a lot of flagging and doing kind of QA/QC, which we still have to do in terms of understanding the LODs, et cetera, and also calibrating the data. So once that's all done, it gets interesting. We can look at the data analyses, and because we are still we haven't totally completed the data analysis, or sorry, the data collection, I'm going to just walk through the processes that we think we're going to do for the data analyses. And I have some preliminary data, but we don't really have very much to show right now. But perhaps in the future, you can stay tuned for another presentation. So as I mentioned before, there's high resolution data, so that's high time resolved data, and low resolution data, which is more for example, the passive sampler, which is essentially an integrated sampler. So again, the high resolution data includes both the daily questionnaire as well as the environmental measurements. We will do kind of summaries of the data and look at what sort of patterns we can see across time. We'll combine the data on these different types of exposures to look at individual level external exposome profiles. And just as an example of that, this is from a completely different study, but you can see we can create different ways of visualizing this, looking at correlations or kind of this type of mapping of different exposure variables to come up with these profiles. So we also want to understand then what explains variation of exposure between people as well as throughout the measurement period. So we can look at, and I'll demonstrate kind of what this looks like. So as an example, here's some of the PM 2.5 data that we collected early on in the study when we first started. So we've broken this out to two different groups, male and female at this time. So we can look at, on average, for all other participants, this was just a few of them, what was the impact of different time of day on the average exposure, as well as location. So we were able to use the UV data to say whether people were indoors or outdoors. And so what you can see is there were different patterns between males and females in this particular group, with males having higher exposures. And what you would have seen before is also, for some reason, there were kind of higher exposures in the evening as well. So breaking the same data down in a slightly different way, we classified them into two different types of employees. At this point, there was only a few in this case, so we'll have more data on different types of employees, but we apportion them to being self-employed or an employee of a company. And so then you can see here that the self-employed person actually had, or people rather, had higher exposures overall. And these were over the day as well as the early evening hours. And then when you actually look at whether it was indoors or outdoors, we see that these high exposures in particular for these self-employed people happened outdoors. So this is an example of some of the querying that we can do of the data to understand better what the patterns are. So another way of looking at the time series data is to try to understand, well, if we looked at the cumulative exposure across the time period, does this happen through short peaks, or is it more of a level exposure over time? And so what we can do is, as an example, here's an example of a time series of data. We can characterize how many peaks there are, what the average peak height is, and how long they last. And basically what we say here is that of the total exposure, about 20% of that exposure happened during these high exposure periods. So of course, what we define as a peak will have to be based on kind of expert input into this. But that's the type of analysis that we can do with a time series. So you can see that we're moving away from just looking at average exposure, which is what traditionally has been used as the exposure measure in epidemiology studies of health effects. So another thing we can do is, now we talked about how we might look at individual level exposure data. So now this is how we look at it, how we might look at them as a group. So once we've characterized the exposure profiles or the exposome profiles of our study population, we can then look at how they are grouped. So we can use clustering and variable selection methods to then apportion these different profiles together and look at how these different groups may differ. So this is an example. It maybe looks a little bit complicated, but we have individual participants on the vertical axis and the different exposures on the horizontal. I don't know if you can see, but essentially through an algorithm, we can look at then how the relationship between the different exposures and then how individual participants might be grouped based on the profiles. And again, we can also select the most relevant variables in the grouping. And this is again an example. So we might find a cluster here. So they have there are similarities in their exposure profiles. And perhaps as an example, this is not real data, but we might say that, okay, these are all people, they're commonly employed in construction and they're less than 50 years old, as an example. So what we want to be able to do is to identify features of these groups of participants with common external exposome profiles. So once we have them clustered, we want to better understand why they have these similar exposome profiles. So this might help us understand the reasons why they have these similar exposures and which exposures tend to go together. So is it that, for example, dust and noise exposure tends to be highly correlated, just as an example. We also want to understand, are there any population characteristics that explain these clusters? So for example, or is it because of specific occupations, age, perhaps where they live? Something I didn't really mention is that we are having participants carry these, do this exposure monitoring, not just at work, but when they're at home. So basically throughout their entire day for about a week. So we can look at both work-related and non-work-related influences on their exposure. Just to give you a sense of the type of data that we've collected through the passive sampling. So I mentioned that this is getting at both VOC exposures to volatile organic compounds as well as semi-volatile organic compounds. So the PDMS sampler is monitoring semi-volatiles, and the 10X will be monitoring volatile organic compounds. So this is a very busy slide, and you don't really have to be able to distinguish it other than to say that we do a screening to determine what the range of different compounds a person might be exposed to. And through this screening, we can then kind of look at whether there are commonalities in the exposures. Now, this is a very busy slide. There will be some where we aren't able to detect them, and there will be some where we are. So as I finish up here, I want to say that we have learned a lot of lessons in terms of just the practicalities of implementing this type of protocol. So it is very exciting because we can measure many things now, and we can measure certain things at high temporal resolution. But this type of technology is requiring participants, for example, to have smartphones. And we did find that not necessarily all have them, and they sometimes can't use them at their jobs. The sensors, again, may not always be easily worn in all jobs. So for example, we had some healthcare professionals who weren't able to wear a watch or some of the different sensors in their job. So we had to find ways around these. And again, just some practical things, which I think we're all aware of, which we saw earlier, software updates and connectivity issues can also pose challenges for using these type of technologies. So I just want to end on so I mentioned earlier that we had interacted with some of you at BOHS on how we could translate some of this technology in the external exposome into practice. So we were interested particularly in the low-cost sensors, or the sensors that we were using, or sensors in general, rather, not necessarily the ones we were using. And we think that this kind of high temporal resolution data could be really insightful for occupational hygiene managers and professionals, because you can get real-time feedback, of course, with the appropriate contextual information that needs to be gathered. And perhaps networks of sensors can also help manage hazard levels across space and time. What you could do in terms of using Millbot technology, such as the app, you can better understand how workers are doing, especially if they're remote or not office-based. And of course, I think many of you probably already use sensors to track lone workers for safety. We had done a survey, which BOHS has helped disseminate, and these are just some results. A lot of people are interested in using these sensors, but they're not necessarily currently used. And you can see, of course, people are most interested in the types of insights that could be provided by sensors. At the last BOHS meeting, some of you will have seen this. So of course, there are barriers to this type of technology, such as financial barriers, not to these technologies, but current methods of monitoring exposure. Financial, as always. The technology we have could potentially increase the number of measurements that can be taken, and to be an effective complement to traditional methods of measuring exposure. I just want to acknowledge the funding source, as well as all the other partners in this project. If you want more information, there's the project website, as well as my contact information. I'm happy to answer any questions. And with that, I will stop sharing, and then I'll turn it over to Manmosij. Hang on a second. If I can do that.
But maybe if I start sharing, it
Yeah,
would stop automatically.
Too many windows.
Yeah, sorry. I was waiting for. It says Mary is still here, but yeah, no, thank you for that, Rhonda. Yeah, Manmosij, if you're able to start sharing your screen.
And do you see my screen now?
Yep,
Oops.
got that there.
There's something over there. Sorry about that.
Looks good.
Okay, so I'll just preface by saying that this is more to do with the method development and things that we've done in E4 project before the sample collection began. So the sample collection is still ongoing, so it's not so much about the results, but more to do with the decision making as to what matrix we collected and why, and what kind of information can we get out of those. My slides are not moving, sorry about that. Okay, so Miranda explained to us about the exposome concept, but I'll be specifically talking about the internal exposome. So mostly the biological responses, be it at the level of the genome, epigenome, transcriptome, or even microbiome. And to characterize that, of course, as we know from human biomonitoring studies, that we need biological matrix to do so. What sets exposome apart from the classical human biomonitoring, or at least it promises, is it theoretically includes all the exposure related changes, be it biological, so from exogenous sources of pollutants, drugs, diet, etc., to other endogenous sources of hormones, microbiome, etc. So what we had to do is to first develop a set of method that could be used in the case studies that Miranda described. So the case study of work packet 6, which is on the internal exposome related to the lung function measurements or lung function decline in the respiratory case study, which is based again on the ECHRS study, which Miranda mentioned already, and then also something that's specific for the shift work study. Now, we have to recognize that in developing or optimizing certain set of protocols, we have to be sure that the choice of matrix reflects the need of the case study. So in this case, I mean, if we are talking about shift work, the biomarkers that we want to study should be reflected as of the chronobiological responses, as well as that of respiratory case study that should be reflected. So we have to be mindful of a few things. That is the study setting or the specific research question or hypothesis that's being included in these work packages, the markers that we have to study and the stability of such markers. I mean, many of these markers are quite dynamic, and we will come to that shortly. But also the matrix required and the ease of sampling, because I think Miranda already showed you the Christmas tree of external exposure. It's equally invasive and complex in terms of biological sampling. So we have to be mindful of all of these while designing the study, the protocol here. The common markers that's being studied across the work packages are hematological parameters, biomarkers of metal exposure, so in urine or in blood samples, inflammatory markers, telomere length, genetic polymorphism, epigenome, proteome. So, I mean, I think you would understand that there are a lot of matrix which as well as a lot of endpoints because we wanted to characterize exposome. But then there are specific ones which are for the case studies. For instance, for work packet 6, we had to take into account the Excel breath condensate and Excel breath VOCs, which Miranda also showed in her protocol. And for work packet 7, because it's related to clock gene and chronobiology, we had to be mindful of the cortisol, cortisone, and melatonin, but also glucocorticoids as part of the HPA axis disruption. This is to show you that, of course, the biomarkers of exposure and effects as well are dependent on the half-life of the stability of these markers, and therefore the time points that we had to choose were also reflective of this. Having said that, I mean, be it in classical biomonitoring studies, I think this a bit earlier, Paramjit mentioned that we should keep our eyes open as to what is known, what is available to observe. And I think we have these information from a lot of biomonitoring studies that have come before the Exposome projects, be it HBM4U or ongoing PARC studies. And there are matrices which can be used, be it hair or tears or saliva, and of course, classically blood samples. The benefit of course, having blood as a matrix is the advantage that it reflects the systemic effect. So often when you cannot have access to the tissue of interest, for instance, in case of respiratory case, you might not always have access to lung-derived samples directly. So lavage or other methods are quite invasive. And that's where the systemic markers in blood are quite well documented, and there are established protocols. But the limitation is it's rather invasive, and when you're depending on the vulnerable population or an age group, even just outside the context of V4, when you want to study them, for instance, in children, asthmatic children, that becomes a limitation to draw sufficient amount of blood to cover all the endpoints. What you can measure in them, almost anything that you want to, and that's the same for the biomarkers as well. So we had to make a conscious choice. We had to think about the question, what if we had access to a tube of blood? What can we measure? And in the coming slides, I'll take you through, of course, the things that we can measure, but also the handling time that is required at the end of the sample collection. So for instance, if we had to do flow cytometry measurement to understand immunophenotypes related to asthma, we would be happy with whole blood samples, but then we need live samples, which has to be stable till it reaches the processing lab. And then the live cells have to be preserved as such. And it requires a handling time of at least 2 to 6 hours, the sample processing itself. But then you get a lot of information of the immune population, immune cell population, as you can see here in the T-SNE plot. Similarly, there are several genotoxicity assays that are available, and we have used quite a lot in the case of health and safety assessment of different chemicals or chemical groups. For instance, micronucleus assay, many of you must be familiar with it. And there also the handling time is, of course, 2 to 6 hours on the day of collection, but then the cell culture procedure takes over a few days. The same stands for chromosomal aberration when you have to understand genotoxicity. And for Comet assay, the hands-on time is at least 2 to 6 hours, and we need live cells without any preservation and has to reach the lab quite soon. So all of these limitations made us choose a certain additional endpoint. So for instance, if you look at the inflammatory markers, they can be rather rapidly fixed within a period of 1 hour or so, and then you can have a multiplex analysis using a very small volume of samples. And the same for the multi-omics. You can do a wide range of these multi-omics, be the starting material DNA or histone. So you can do a lot of epigenomic analysis, microRNA or mRNA. You can do transcriptomics and proteomics, and the hands-on time is less than an hour. That's all great when you have availability of this sample. But what if this doesn't happen? Or what if you don't have access to it? You don't have the hands-on possibility of collecting a sample at a distant site or a factory where the samples cannot be processed. Or for our case, which was COVID, I mean, that motivated us to look into a bit more into the non-invasive or self-sampling methods, where you don't have access to phlebotomy blood, but you could work with other sources. So what we did was we looked at the literature. We did a in-depth review of matrices, which can be non-invasive and self-sampled. So for instance, urine, feces, saliva, breath, sweat, amongst others. You had potentially applicable but minimally invasive, so blood using finger prick or some other methods. And then there are on the side, you have different matrices or different endpoints that are going to be measured in the E4 project. So that includes metals, cotinins, polyaromatic hydrocarbons, BTEX, and then amongst the biological markers, immune factors, cortisol, melatonin, and so on. And you would see that urine is one of urine, saliva, breath, and a partly non-invasive sampling of blood allows us to address all of these in a self-sample way or in a less invasive way. So from here on, we wanted to move forward with the matrices that we chose. So XL breath condensate, XL breath VOCs, blood using phlebotomy where possible, or dried blood spots, saliva, or urine samples. Now, once we have these samples, we also need contextual information, which Miranda mentioned. That includes, of course, the exposure assessment, but also the questionnaire information that's collected by the study protocols in the respiratory health, but also the shift work. So we combined this and wanted to see in terms of setting up the SOP, we wanted to test out the storage and collection workflow, but also the feasibility of this sampling. As you can see here, the biological sampling that we described takes at least an hour, other than the questionnaire itself. So VOCs over 20 minutes, XL breath condensate over 15 minutes, spirometry 5 minutes, and then different blood sample collection 10 minutes, and finger prick and saliva. And so this is a customizable workflow and is defined in an order where the last endpoint is affected least by the previous endpoint. So for instance, if you were stressed or something because of blood sample collection, the XL breath VOCs might be affected, and so would the XL breath condensate. And so that's why we wanted to prioritize that as a first one. And then the urine samples could be the last or the first one, irrespective of, and depends on the volunteers when they can provide the samples. We also wanted to address the fact that many of these markers are highly unstable. So there are temporal variations depending on when you are studying it, be it a winter or summer and so on. So we collected at 3 time points, each of them 6 months apart. We provided a detailed instruction as well as a video protocol to the individual participants on collection of urine samples. It was rather simple, a document instruction of saliva, and when it was a more complex process where they had to do a finger prick, we gave them a video protocol which they could follow step by step, and that's available on our E4O website. So we before asking actual participants, we had to ask for the ease of sampling to the volunteers. So the first question was the clarity of the instruction, which was relatively clear for all of these sampling methods. But in terms of the ease of sampling, where it's very convenient to not convenient, you would see that urine sampling and saliva sampling were rather convenient for most, while dried blood spots were less convenient, but still we received. I have to say that of all the participants, all the 40 participants that we recruited, we received all blood spots in good conditions, even though they were less convenient, but we received them all in good conditions back. And this was done at a home setting of the individuals, and they shipped it by postage to us for further processing. Of the samples that we collected, now let's see what we can measure in those. So we can have from all of these markers, genome-wide studies, so polymorphisms, DNA methylation studies, both targeted and untargeted, the volatile organic compounds in the cell breath condensate, the immune panel, so that you could expand it based on whatever platform you're using, but that's quite customizable. In urine, you could analyze the polyaromatic hydrocarbons, cotinins, alpha amylase in the saliva, and melatonin and cortisol, of course, and then untargeted proteomics in these matrices as well. So this workflow was perfectly tailored for the E4 case studies. I have to give you a fair set of warning that this is not something that you can just take as it is into another exposome study. For instance, if you go to the EHAN website, you have 9 projects, and you will see certain protocols have been tweaked a bit to fit that particular study and the end results. Now, in the optimization of the dried blood flow collection sample, so this is a result from one of our partners from Karolinska. Here on the right side, you will see that the serum markers of the different cytokines and chemokines correlate pretty well with the dried blood spot sample. So that shows you that in a scenario where patients or samples are not accessible, or you don't have enough a workforce to collect samples, a nurse, for instance, you could easily ask participants. And this is something that we have expanded into in case of minors in Congo, where we have received samples in good conditions, and we are going to start analyzing these samples to see what kind of results we receive out of it. The DNA quality and the yield is rather satisfactory for us. So from the dried blood spot as well as from the phlebotomy, of course, the yield is a little lower, but we have received it in very good condition and are of good quality and can be used. We've been able to measure the EBC quality. So if you look at the saliva, there is alpha amylase, which is rather high and which is expected. But if you look at the EBC collected from the same individuals, they were rather below limit of detection, which is desirable. So which means that the cell breath condensate collection works perfectly well without contamination from saliva. So when you're looking at the markers, they are not contaminated from another matrix, but very specific to the EBC. We were able to get good quality results from the plasma and the EBC cytokines as well over the 2 different time points. So you will see that most of the cytokines are comparable over the 2 time points, so which was quite good in this case. And when we look at the correlation between the EBC and the plasma cytokines, we do not see often a very good correlation, which in a way is good, because we are in case of the EBC, we are looking at the upper airway inflammation, and we are looking at inflammatory signature, which is from the lung, while in plasma, we are looking at systemic effects, so it could come from anywhere. So this gives us good quality results. We also looked at the cotinine levels to account for the smoking habits. So you would see that our heavy smokers are represented here with the red dots. This dot corresponding to somebody who is 105 cigarettes per week, and then here somebody with 10 cigarettes per week. So this also allows us to, in this semi-invasive, non-invasive workflow, allows us to correct for these factors. When we move on to something untargeted, we did untargeted analysis of the proteomics of the EBC, and we find clusters of keratin and other proteins which are representative of the processes such as apoptotic or cytoskeleton organization or DNA repair processes. And when we take the asthmatics, I think we would have very interesting results to explore there as a relatively new matrix. So what we have so far is a SOP that you can easily use. It's made available online through our website. It's a written protocol with a clear information on the workflow of what temperature storage condition works best, what kind of tubing and everything that you need, what products. And if you look at the blood collection workflow, this is further subdivided into several parts where different samples are processed and for different endpoints. And this is also part of our WeExpose toolbox, which will be continuously updated. So as soon as we have more results, we will update the protocols, and this is a living document, so to say. So with this, I want to thank these happy faces from the EFAR project, and also we are part of the larger European Human Exposome Network. So you might want to check that website and see what other partners are doing in terms of characterization of internal exposome. Some using relatively well-established protocols, other developing, like for E4, we are developing custom protocols for the characterization of occupational exposome. So with this, I end my presentation. Thank you.
Thank you so much, you two. My mind is blown. I don't know about anybody else, but wow, there's a lot going on with that. It was really interesting. Thank you so much. So we have enough time, 10 minutes or so for Q&A. One question's come in so far, but please, you guys, if you'd like to put any more questions in the chat box. Also, feel free to raise your hand, with the little button right there, and you can always speak your question as well. So yeah, Miranda and Manoj, if you're ready. So the question that we have aligned is the E4 approach with the lifeline exposure model approach developed a few years ago now, and the learnings and outcomes of that. Noted also the complexity of the long-term or lifelong nature of whole body exposures and disease initiation progression, including radiation, dietary intakes, and contaminants therein. Who's either Miranda or Manoj? Oh, there you go.
So the question was, how well aligned is Exposome or E4 with the lifeline model? And then what was the second part?
The complexity of life-term nature of whole body exposures and disease initiation progression. If Alex is on, if Alex, you're very welcome to ask the question in your own way, just so I make sure I'm saying this right. If you wanted to unmute, but up to you. No, it's okay. Well, how about if you just go for that first part, just E4 is with the lifeline exposure model.
Do you want to go, Manoj, or?
No, I mean, I think if I'm not mistaken, this lifelong exposure, it's a multiple stressor, something related to environmental pathways, and I think it was part of Helix or Heals, some other projects, but I'm not very familiar with it. If I may add, I mean, the questionnaires that we include aims to collect a lot of those information, I mean, in terms of the lifelong exposure, because we are looking at a cross-sectional study design, especially in this short period. A lot of it relies on reconstruction of exposure through job exposure matrices and other. So there might be error in the recollection of these workers, but I mean, that's the best we can do in terms of gathering information on their past jobs and so on. But Miranda, if you wanted to add something on that.
So yeah, I'm not familiar with the lifeline exposure model, but if it is what Manoj just mentioned, I mean, yes. So in our project, we have the cohorts that we derive these studies from. So the two case studies we've mentioned, they will have more data that was collected through the cohort. So they have been ongoing for some time, and some of that may include exposures previously. For previous projects on the exposome that I have either worked on or more familiar with, it is, you have to reconstruct, and it is difficult because, you know, you don't always have data for where, you know, the different places people were before, and also you have to know where they were at all points of their life. So their residential history, their job history, and things like that. I'm not sure if that answers the question.
If we Alex
Yeah.
commented very true. So hopefully I think so. Well, just for time, if I move on, there was a couple more questions that just popped in. So another one from Roger is, are stress factors or mental health considered in the external exposome?
Yes, it is something. So Manusha mentioned that they are measuring stress-related biomarkers. Do you want to say something about that, Manusha?
Yeah, so there are some mental health related questions especially in the shift work case study, not so much in the respiratory. And I think the questionnaire is available in the Expose toolbox already. However, it's not such an in-depth questionnaire of mental health. There's another project I'm aware of within the EHAN network that's really focusing on mental health as one of the stressors or external factors. Yeah.
Okay. No, thank you. Actually, I had sort of a similar to that ties in. Have you guys heard of something called, I'm sure you have, Fitbit anxiety? So you know what I mean, when people wear Fitbits and they don't hit their targets for the day and they get stressed out and think, oh, I'm gonna, you know, my health is gonna get worse. And I suppose it kind of feeds in, or it could feed into in a similar way to Exposome. What do you guys think about that? Could there be an Exposome equivalent of a Fitbit anxiety to anticipate, or?
Well, you may know about, there is always that if you ask someone to carry a measurement device around, do they actually behave differently? So that is an issue. But because these people are carrying things around quite a lot, they get used to. So people do get used to that at some point, oftentimes. So in a previous study that I've done, they actually got real-time feedback. So in this study, they don't really get the real-time feedback. It's the collection of data is primarily for research purposes. So it's not like if you wear a Fitbit, it tells you you've done however many steps today. There is this possibility that people adjust how they behave because of that. But generally speaking, oftentimes if people are in studies long enough, then so the type of study we do, we don't necessarily give them the real-time feedback because that's not the point of the study. But if we were doing a study where we're specifically looking at that as an intervention, we might look at that.
Oh, I gotcha. Okay, that's fair enough. Another question here. So can you already estimate when the studies will be finished and the results evaluated? And is it going to be published or otherwise made available for professionals?
So I believe that the data collection should be stopping this year. I'm not sure exactly when, and then we will anticipate maybe another few months or a year to analyze the data. I mean, with research studies, you can have endless analysis of data, but the project ends, it doesn't end in 2024, as I think I had in a slide, because there's been an extension to 2025. So we do expect that we will have results within a year, at least some preliminary results, and they will all be published. So they will be accessible to other people.
Excellent. Great. Well, no other questions in the box here, so I may maybe give everybody another minute just to scramble a question, but if not, then we can, you know, knock off 5 minutes early. But a really excellent presentation from all three of you guys. It was just interesting how it actually kind of coincidentally fed a little bit into Parm's theme as president-elect as well. So that was, I think it was actually quite well put together. The good timing here, huh? Any comments from you, Parm?
Yeah, excellent. Thank you very much, Mary. I thought that was an excellent transition of looking at historical issues and applying modern science and technology to advance our understanding and thinking. I just wish I was as clever as our other two presenters this morning. Puts me to shame the work they're doing.
Oh, gosh. No, it's really, really wonderful. We're getting just flooded with thank yous and applause and things here on the chat box. So you guys are very, very much appreciated. And yeah, no, thank you again, all three. I don't see any further questions, but I'm sure you guys would be happy to be contacted if anybody thought of a question later. And yeah, thanks everybody for joining. I thought that just excellent. Yeah, for all your time and effort. It was very much appreciated. And yes,
Thank you
see
for
you all
hosting
soon.
us.
Yes,
Yeah.
thank you very
again.
much for inviting
Thank you
us.
very much. Yeah, of
And
course,
thank
of
you,
course.
Mary, for the invitation and the sterling job that you always do to
Oh,
organise everything.
goodness. No, you guys make it easy. It's fine. No, it's easy.